Yeah.
No vamos a sí.
Uy, aquí está.
Hola. Hola a todo el mundo. Hello everybody. Thanks for coming here and thanks to ECUSA to organize this event. We are going to start this series or talks organized by ECUSA here at Georgetown University. And the first one is about neuroscience. So the speakers that we're going to talk today, they are Sandra Jurado. She's here. She's assistant professor in the Department of Pharmacology at University of Maryland. And she's going to talk about the adaptation of the neuron circuits during neurodegenerative disease. Then Regina Rincon-Torroella, she's a postdoctoral fellow in the Department of Neurosurgery at the Johns Hopkins University. And she will talk about the intraoperative detection of brain cancer infiltration in optical nerve. And then Javier Traba is a research fellow at NIH, and he will talk about the regulation of inflammation in caloric restriction. Right? So, and I am Sonia Villapola. I am assistant professor here in the Department of Neuroscience at Georgetown University. And I'm going to talk about the restoration of the damaged brain. So, as you know, in this country, around 3 million people, they have some kind of the brain injury, either as a result of trauma, a stroke, brain tumors, or any kind of infection. So the deficits of these injuries, they have several consequences, that is such a physical problems, some sensory deficits, behavioral change, and cognitive and communication deficits. So, Santi, please, can you throw the light? Yeah. The light. Las luces puedes apagar. Yes. Gracias. Oh, yeah, much better. Thank you. So this kind of injury has several consequences. So that depends on the type of the injury. But in general, all them they are characterized because there are a consequence of the primary injury as a mechanic damage, and then in the following hours, days or weeks, it occurs a secondary injury that can cause a blood barrier breakdown, cell death, inflammation, tissue damage, and all this process, like cytotoxicity. So the goal, in general, the goal of the share is restore these brain regions affected by secondary injury. So there are not, right now, there are no effective treatments for this kind of brain injury so far. So we need to find new drugs or new treatments for this secondary damage. And for this purpose, we need to use some animal models in the laboratory to reproduce these injuries. In my career, I have used these 3 animal models that you can see here, some kind of representation in rodents, that there are the cytotoxic injury, ischemic injury, or the traumatic brain injury. So all them, the 3 of them, we have some kind of consequence very similar, that there are kind of the secondary brain damage. And these that are characterized by the very fast neuronal death, as you can see here, apoptosis, astrogliosis, inflammation, and or angiogenesis. All these factors, they are in common, all these kind of models. And I would like to start with talking a little bit about the ischemic injury. And as you can see here, this is the representation of the brain cells of these animals. This is there are pups, rats, that there are the 7 days old. So we can generate an ischemic brain region on these animals, as you can see here. And then there are characterized for this massive cell death, as you can see here in these cells that they are dying. This is the green cells, and also inflammatory response around this area. That is the you can see here in the red color. So all this inflammation can occur days or hours after ischemia. So, but as you know, these studies were performed in these immature brains. And as you know, the babies that are not treated adults, so the treatment that we need to look for the adults injuries, they are not the same that for the immature brain. So who we can protect the premature babies or from this kind of injuries, from brain damage. So we start to study some kind of the treatment that about the melatonin. So as you know, the melatonin is a hormone that can regulate the circadian rhythm, and is stimulated during the night, so that helps to sleep. And the babies, but the newborns, they don't have this rhythmic secretion of the melatonin. Is the main reason that they don't sleep very well the first days since they work. So, but there are a few studies in vitro that they show that the melatonin they have another effect that is can be anti-inflammatory and neuroprotective. So, what effect does the melatonin have on the brain damage in these immature brains? So, we study in our animal model this compound, and we demonstrate that administration of the melatonin in these neonatal rats, this rat 7 days old, after ischemia, could increase the myelination, and these areas that there are characterized to get like a strong axonal damage after ischemia, that is therefore corresponding with the white matter. And so the melatonin treatment increased this production of this melanin, and also the number of the mature oligodendrocytes that there are the cells that produce the melanin. And so we can think that it could be the melatonin could be a good candidate, a good neuroprotective for the candidate for the premature babies. In fact, years after our investigation here with this compound, several hospitals in France, they start to use the melatonin in clinical trials to treat these premature babies. They have some kind of injury in the white matter. It seems that the babies they were recovering. So another compound that we're studying was the inhaled nitridosis. The nitridosis, you know that when the premature babies they born with some kind of the problems with the lung development. So they inhale the nitridosis would prevent, will help to this lung development. So was used like a systematic treatment for these babies, for these premature babies, but was not associated with some detrimental growing incidence of brain damage in these premature babies, but we don't know which was the effect of this nitric oxide on the brain when there are damage. So this is the main reason that we start to study this compound in more animal model. And we found, surprisingly, that the inhaled nitric oxide was neuroprotective in these animals, and after ischemia in one model. But when the inhaled nitric oxide was administered during ischemia, when it was administered after ischemia, like hours after or days after, was very detrimental. So it was not good for the brain, can cause huge hemorrhage in this baby. So the time when the inhaled nitric oxide was administered was very important in our studies. And also the dose, the concentration, only one was the dose was 20 ppm, so parts per million. Only was at this dose when was good. So in general, we have to think that everything, and neuroprotection and to restore the brain, everything depends on the age, the timing, and specifically about the dose. And what happened with the gender. Also, the gender can play a role in neuroprotection and to repair the brain, because we know that the females and the males, they have the brains that are different. So, but are the males or the females responding the same after brain injury? We don't know. And our preliminary data or field studies you show that the females, they have a reduced inflammatory response in our animal model of traumatic brain injury, and the microglia activation was reduced in females compared with males. So, and that occurs very short time points after injury. So, what kind of treatments we can apply to this kind of injuries? So far as you know, there are no specific treatment for traumatic brain injuries or concussions, as you can see here in this football place. So, but we start to think that we know that the hypertensive people was treated with is treated with angiotensin receptor blockers. So we also the receptor, these blockers, the drugs, they have some antioxidant and anti-inflammatory effects. So we are worrying, we're thinking that maybe these sometimes these drugs could have some role on the injury brain. And we use our animal model of TBI, and we injure these mice, and we administrate this kind of these sometimes these angiotensin receptor blockers. And we perform some studies, biochemical histology, behavioral studies, and we found that these animals they were treated with these both sartans, they show like a reduction in the lesion, very low dose compared with high dose, and also the time the therapeutic with the Western standard anti-seizures was very promising, and the pathway, the mechanisms these drugs were working were through the PPR activation, one of them. And finally, I would like to make the next question: if the damage in the brain affect only the brain or also the rest of the body? So the injuries in the brain can affect more than only the brain, can affect different organs. And as you can see here, we know that the trauma, injury in the brain, can induce or release some molecules, some cytokines, or some pro-inflammatory factors that are transported into the circulation. And at the same time, the liver, in response, produce acute phase proteins. And these proteins, such for example, the serum amyloid, can be released into the circulation again and can induce more pro-inflammatory factors that can affect the damaged brain getting worse. So that was some interesting question that we want to reproduce in more animal model, TBI. And we demonstrate that this phenomenon and showing an increase of the liver levels of these molecules, these proteins, and the animals a few hours after injury, and they are also the circulation, or this molecule was released in the circulation. So, surprisingly, also we saw that this molecule that was produced by the liver, was released by the liver after injury, will affect inflammatory cells in the brain. So, will be like a reverse effect. So, and then just to finish, just to say that, in conclusion, we can say that to restore and repair the damaged brain, the neuroscientists, we have to think outside the box. That's mean we have to think that the immature brain is not the same as the adult brain. It's working different. The plasticity and the immature brain is in all the features that are different. The female brain is not the same as the male brains. And a different dose of something doesn't have the same effect. And the tiny administration also does not the same effect. And different genotype is not the same effect. So the most important is that the damaged brain doesn't mean that we are a healthy body. So I want to just say thank you to all the academic institutions that support me all these years, and also the people who I have worked with over the years, and thank you everybody to be here. So now if you want, Sandra can present her work, and then we can go to the questions.
Okay. Well, thank you very much, Sonia, for the invitation. Thank you, ECUSA, also for organizing this series of events today and in the future. And for all of you being here in this snowy, kind of snowy day. So today I wanted to talk to you a little bit about the work that we are doing, one of the lines of the lab. And it is about verifying adaptations of neuronal circuits during neurodegeneration. So neuronal circuits, as this one here that is beautifully drawn by Cajal, the one in the hippocampus. It's an important region for learning and memory, and it has different neurons that are connected. There are different neurons connected in a different way. And this drawing from Cajal, of course, is just representing kind of like a snapshot in time. This is how it was the circuit at that time, but over time we are learning, and we have learned actually, that circuits are not rigid, are very plastic and very flexible structures. And they can be plastic in different ways. The strength of the connections between neurons, they can become weaker or stronger depending on experiences and the activity patterns of neurons. And this is a phenomenon that we call synaptic plasticity. And it's been studied actually in the hippocampus, really see here. But also with the improvement of imaging techniques like 2-photon imaging, we also over time we actually see that the brain can change physically. Even right now in our cortical neurons, and I'm sorry this is showing like that, they are just undergoing some plasticity that involves changes at the physical level. Here we have a cortical neuron that is being imaged with an in vivo 2-photon technique, and then we can see this is changing constantly. This is actually a time lapse of 30 days. So all of this plasticity we think allows the brain to encode new memories, blending memory. Of course, this is something very important. But having this ability of change can produce some problems as well. Sometimes the brain, instead of adapting in an effective way, it can produce some kind of maladaptations to sort of the stimuli that it receives. Very clear example of that is in the case of drug addiction. For instance, the brain is exposed to cocaine, and the brain produces some adaptations to cocaine that in some individuals can lead to drug addiction. So this would be like an example of maladaptation. The brain, and actually this type of aberrant plasticity is being studied heavily in this type of context, like drug addiction, but not as much in the context of neurodegeneration, for instance. We think in diseases as Alzheimer's disease more in terms of cellular and molecular levels. My lab is interested to look a little bit beyond that. Actually, Alzheimer's disease is characterized traditionally as these accumulations of metabolites like these amyloid plaques or tau tangles that produce brain atrophy. And this is a cellular and molecular changes that happens in the cells that usually they become apparent kind of in middle stages and later stages of neurodegeneration. So in the lab, we're a little bit more interested in actually looking at something that happened earlier, and some of the symptoms can indicate that maybe a neuronal circuit is being kind of damaged, representing some kind of like deficit in function very early on. So we start to look at, for instance, early symptoms, and we see, we all know that memory loss is very one of the landmarks of Alzheimer's disease. And actually, very early on in this phase, we see actual memory, neuronal loss from cortical region that is called the entorhinal cortex. And we can look at this, well, not in my lab, but in other labs that use functional imaging. And also, another part that is affected very early is this loss of function. And of course, in humans, all function is not key for our survival, but actually it can have a profound effect in these patients because, well, they are not attracted to food in the same way, they lost appetite, and they actually, these people become weaker. It makes the whole spectrum of the disease. So these two early symptoms, these two early evidence suggested to us that there was maybe this adaptation, an early adaptation, maladaptation of a circuit that is formed by the olfactory bulb and the entorhinal cortex. That is this pink blob that you see there floating around. So we wanted to understand and study how these circuits may be changing and telling us something about earlier stages of Alzheimer's disease. So what do we know about this circuit? Well, we know that the olfactory bulb is connected to this other intermediate region, the piriform cortex, and that from the piriform cortex, there are some projections into the entorhinal cortex that is actually projected into the hippocampus, this region that also and is important for learning and memory. But in the lab, we're also very interested in understanding direct projections from the olfactory bulb and the reciprocal projections from the entorhinal cortex to the olfactory bulb. So this is the study that we wanted to do and to look at this over time in naturally aging mice, but also in Alzheimer's models, animal models of Alzheimer's disease. But as you know, you can imagine, just looking for connected cells and different types of circuits in the brain is a big challenge. Actually, there are multiple initiatives right now in trying to solve this problem and trying to form clear maps of connectivity in the brain, with the idea that we will be able to look at how these neuronal circuits adapt. And this is, of course, because the brain has a lot of neurons something like in the range of billions of neurons. It's been calculated, suggested that we wanted to count every single neuron in the brain individually by hand. It would take us like something like 3,000 years. And then from, after all that amount of time, then we will have to figure it out how a cell is connected to what. So we don't have that amount of time in my lab, in my life, and the NIH doesn't have that amount of patience for us to try and figure it out. So we need to come out with ways to actually make this like a really faster and more efficient. And this is some of the techniques that we are implementing in the lab and also optimizing that help us with this challenge of understanding circuits and adaptations of the circuits. We are using trying to optimize these viruses, these rabies viruses, that they can actually be modified. We can modify the genome and make them so we can use them to track neuronal circuits. So the rabies virus is infecting one neuron, and actually it jumps into whatever cell is connected to the cell that we infected, and only to those ones they will jump in a different way. So that way, the rabies virus is able to infect the whole circuits in the brain. And to make this workable in the lab, because rabies virus is very toxic, for the test indicated these techniques, they replace one of the glycoproteins, the gene, the genome, for something else that is less toxic, like a really progressive protein. So we actually can detect what cell is infected and what cell is projected to that cell. And we can use GSP or anything else that we want to express in the protein genome. So the way that this works is basically we can inject, let's say, the olfactory bulb with the rabies virus, and that is going to exchange the GSP. The rabies virus then is going to jump one synapse, one connection, and it is going to instead whatever neurons are projected, some of them are going to be in the entorhinal cortex. And because of the modifications that this rabies virus has, actually this is only going to happen once. It's going to be a monosynaptic tracing. So using this technique, we are able to actually, in combination with different proteins like different fluorophores, we can start creating maps of connectivity in the brain. So by injecting in different brain areas with different colors, we can see different patterns of connectivity. Sometimes the overlap, sometimes it's different, and we can start to identify the neurons that are connected and study the properties. So this is the type of work that we are doing. Basically, we just started and we are injecting these rabies viruses in the olfactory bulb, as you can see there, and then we can identify the neurons in the entorhinal cortex that are expressing the GFP. So we can start asking questions about the synaptic properties, the synaptic plasticity by using electrophysiology. Basically, this is like overview of synaptic properties in a naturally aging mice. Of course, we will apply this in Alzheimer's models and trying to identify exactly what are the changes that happens in this network. Another interesting feature of these neuronal tracers is actually they encode proteins, they incorporate genome, so they can be used with pre-transgenic mice to identify particular cell types. For instance, in the entorhinal cortex, the ones that maybe are more fragile or more susceptible to disease. So, of course, not every single neuron in the entorhinal cortex is going to be the same. They're going to be different types, and some of them we expect that actually they are going to be more susceptible. They're going to have different molecular profiles, and by incorporating and identifying these molecules, we hope to identify biomarkers for early cortical neurodegeneration. With that, I just want to finish, time to say, of course, I thank you to the funding agencies that funded the lab, and Sonia for the invitation, and ECUSA for the invitation, and all of you for coming.
Okay, this is always the most difficult part, getting everything set up. Thank you so much for the invitation here, Sonia and ECUSA, for being all here today. I want to make this very easy going, just to explain a little bit why and how I'm here, and a little bit of the research that we are doing. So the brain is the unexplored frontier, the same as the universe, and it's the passion for the brain that drove me to be here today. And everything started in the University of Barcelona, where I studied in my medical school. I got my MD there. And this is a laboratory of surgical neuroanatomy, where in a very humble environment, I found my passion for neurosurgery in the cadaveric simulation lab, where we did simulations of neurosurgical approaches to the brain. And the idea of becoming a physician scientist kept me from sleeping at night. So that's how I ended up willing to come here to the United States and train, because I thought that the vision of the surgeon scientist or physician scientist was much more stronger than what happens in our own country, Spain. And it was that very same university where Santiago Ramón y Cajal spent 4 years as a full professor, starting his research that he himself claimed that helped him achieve the Nobel Prize later in 1906 with his friend Camilo Botti. And I put this slide first because I love Santiago Ramón y Cajal and his work, and second because Sandra challenged me and told me that there's no good Spanish neuroscientist who doesn't put a slide of Santiago Ramón y Cajal in his presentation. So I got a grant from La Caixa that allowed me to start my postdoctoral fellowship in between my medical school and starting residency in neurosurgery. Life wanted me to go that pathway with Dr. Alfredo Quiñones Hinojosa, and I feel very fortunate of being able to work with him. And he likes to claim that he is my mentor and also my tormentor, because he was featuring nature for his hard-working mentality and the 24/7 lab spirit. And trust me, that's how he works. So I want to describe a little bit that work that we do there. And for me, he is the typical great example of a physician scientist here in the United States, especially being a neurosurgeon. What he does is he takes advantage of everything what he does, from the OR to the bench, back to the OR. And in that sense, in the lab, we do a very big variety of science, all the way from clinical research, analyzing the outcomes in our patients, to basic science research using the same material that we extract from the patients and would go to medical waste to the lab to find new models to fight brain cancer. And then at the same time, using all the patient surgery and work in the lab, we work in academic work to be able to explain what we are learning to the other people. And in that endeavor, I've been able to work in the new video atlas of neurosurgery, where we are putting together several videos to show to residents and young attendings how to do the surgeries. And it's going to be the first book of this kind. And we are finishing it, but we better rush because it's already selling in Amazon. So the part that I would really like to concentrate in my talk is the work that I've been very fortunate to do in translational research together with Dr. Quiñones, Dr. Shinde Lee, Tyson Chaichana, and Carmen Kat, that started in this work in collaboration with biomedical engineering, where we have designed a laser that is allowing us to see cancer versus non-cancer visions at the operating room while we are doing the surgery. And why is this important? Glioblastoma is one of the most devastating cancers that exist. The overall survival of a patient with glioblastoma is of a median of 14 months, no matter what. We can do whatever we want. For the last 30 years, only 4 treatments have been approved, have been FDA approved for this type of cancer. And even Walter Danby, who worked at Hopkins, the same place where I'm lucky to be right now, did some research more than a century ago, where he took the whole hemisphere of the brain of the patient to try to battle this cancer. And the cancer would even recur months after that at the other side. So this is the pathology that we are trying to find. What we can do as neurosurgeons nowadays. So the only thing that we, with our patients, have found that can help increasing that survival is the extent of resection. So how much of that tumor we are able to take out. But always trying to preserve the patient. How can we maximize the resection without compromising the neurological function? So there's a lot of methods that have been studied, but the problem is that as a neurosurgeon, once you are there, the tumor looks so similar than the normal brain that there's no way that you can easily distinguish it. So you are fighting in between how much I'm gonna take. Is that gonna be tumor or is that gonna be the patient's speech? So we work with biomedical engineering to use an optical laser that the ophthalmologists have been using for years to look at a histological resolution at the back of the retina, and it works as the ultrasound. So it's saying but instead of sound, it uses a light wave. So it sends a light wave, infrared light, and it uses the absorption, scattering, and reflection properties to compare the optical properties. So first of all, we wanted to know if that would work in telling us if we were looking at cancer or non-cancer tissues at the resection margins. So we tried with biopsies of Dr. Quiñones patients, and we found that indeed the cancer had different optical properties than the normal tissue. So with different complex algorithms that biomedical engineering built, we were able to obtain these maps, very easy for surgeons, red cancer, green non-cancer, that would be able to guide us through the resection during the surgery. We would go there, take the tumor out. When we are in the margins and our eyes are not able to distinguish in between cancer and non-cancer, just move the laser around and know if we are facing cancer or not and taking it out. The first was trying getting biopsy samples ex vivo with the Dr. Quiñones patient. The second was trying to see if we could move that to the OR, to use it in the operative room in our patients. Before trying it in the patients, we did it in our mice model of human brain cancer, where we take the glioblastoma for the patient, we grow it in the lab, we implant it in the mice, in immunosuppressed mice, and we grow exactly the same tumor that our patients have in that mice. And what we did is, we did a surgery in the small mice, and we used the laser to detect if we could move that to the OR. And this is an example. You can see the laser there, the mice is breathing, so it's live surgery. We took it out, and we see there not only cancer versus non-cancer, but also in a very high resolution, where are the blood vessels, where it's very important to avoid stroke. And here you have an example, these micrometric vessels of the mice that can be seen with this new technology. So once we knew that this was a reliable, non-invasive, non-contact, non-radiation, and fast technology, we wanted to try to move it to the patients. And as you know, here you have to go through all these IRB approvals, all the committees, but finally we got the approval and we tried it with 3 different patients in the first clinical feasibility study that we had for this project. And this is the first step, but as Albert Einstein said, no problem can be solved from the same level of consciousness that created it. So we know, as Walter Dundee told us more than 100 years ago, that no matter how much tumor we can reset, we are going to cure these patients. So the next step, what we are studying in the lab, is the basis of the brain cancer and how we can fight it. I don't know if we have enough time, but I have a 5-minute video that explains a little bit what we are doing. And this is the second part. So since we cannot do that with surgery, how can we find a cure that finally eliminates this part?
My team and I have a unique mission to provide the best possible health care to those diagnosed with brain cancer or brain tumor. We strive every day to try and find a cure for brain cancer. Our first priority is to successfully remove as much of the tumor as possible and alleviate the concerns of each of our patients and their loved ones. We then focus our attention to finding a way to increase the lifespan of all our patients diagnosed with brain cancer. We dream of one day that brain cancer will no longer be a cause of death for so many. Over the years, I have been developing a team of physicians and scientists from around the world who are dedicated to transforming this dream into a reality. Throughout all my years of experience as a physician and researcher, I have found that creativity, passion, dedication, and teamwork are the best tools used in this fight against brain cancer. I strive as a mentor to encourage my students to be passionate and compassionate leaders in healthcare and science today. At Johns Hopkins, I am the principal investigator of the Brain Tumor Stem Cell Laboratory. This laboratory is one of a few neurosurgery laboratories that have received funding and support from the National Institute of Health, a federal organization dedicated to supporting research. Within my research team, we are exploring many different areas of brain tumor research. One of the main focus in our laboratory is the understanding of the mechanisms of cancer. The researchers in my team focus on the most devastating types of cancer, one of them being glioblastoma multiforme, which affects humans. It is imperative to invest all the time, effort, and resources to develop new techniques to combat this devastating disease. We want to be a source of hope and courage to all of those who are fighting for their life. And we want for them to know that every minute of every day, someone is trying to find a cure for their disease. It is only now, after all the effort we have invested in generations of researchers, that we finally are beginning to have a notion about how brain cancer develops and how it acts. However, we have a long road ahead of us as we strive to find a cure for this disease. One of our ongoing projects focuses on stem cells harvested from human fat as a brain cancer treatment. We use it as a Trojan horse. The heterogeneous makeup of brain tumor makes it near impossible to destroy with current therapies. Mesenchymal stem cells isolated from human fat tissue have the potential to act as a smart device that tracks migrating brain tumor cells. We can then use this smart device as a Trojan horse to deliver a knockout punch. This incredible ability can be harnessed by modifying these mesenchymal stem cells to release a therapeutic molecule. In this project, the therapeutic molecule is BMP4, bone morphogenic protein 4, which has been shown to make brain tumor cells grow less, migrate less, and even be less aggressive when it comes to cancer. Additionally, mesenchymal stem cells open the door to personalized medicine. Patients can donate their own fat tissue, where researchers can then harvest the mesenchymal stem cells, modify them, genetically engineer them if necessary, and then use these mesenchymal stem cells from fat to fight their own brain tumor and maybe in the future fight any other neurological disorders. We need continuous support for this research, as we hope to be able to keep making progress in this fight against brain cancer. We cannot do this alone, and we thank you from the bottom of our hearts and on behalf of all those patients and families struggling every day against brain cancer. We thank you for believing in us, and we thank you for giving us hope.
I'm not gonna do it, but now is where I ask you to donate. Well, that's a little bit of summary. And as we try to do that, I think that one of the most important things is the mission of ECUSA. Try to build bridges instead of creating borders and impulse, right? And I really want to thank you for being here, for inviting me, and thanks to all the team for all their amazing work. Thank you.
Thank you, Sonia. Thank you for the presentation. So the title of my talk is Regulation to Inflammation by Calorie Restriction. Let me give you first a little bit of background on what I did before I came to the United States. It's more related to neuroscience than what I'm doing now. So, as you can see here, we have area. The second signal is transmitted from the neuron to the axon to the terminal. And here is like assuming to be a synapse and connection between the two neurons. So the first synaptic neurons, when the nerve signal comes, releases a molecule into the synaptic cleft called neurotransmitter. The major neurotransmitter in the central nervous system is glutamate, and glutamate activates some receptors in postsynaptic neuron, and this transmits the signal to the neuron. So the concentration of this neurotransmitter glutamate is kept very, very low in the synaptic cleft. This is very important to keep the signal under control. This glutamate is kept very low by the presence of glutamate transporters, both in the presynaptic and the postsynaptic neurons, but especially in the astrocytes that's pathological conditions such as ischemic fusion or brain trauma, neurons lose the capacity to take up the glutamate, and thus the glutamate accumulates in the synaptic cleft. This is because these transporters require energy, and in ischemia, they lack energy to perform the activity. Also, sometimes they are in reverse, so the glutamate accumulates several whole levels in the synaptic cleft. Here we have a soup of what happens inside the postsynaptic neuron. Glutamate will activate NMDA receptors and other receptors, which will cause a huge increase of calcium in the cytosol of the cell. This calcium is still performing its normal functions to be transported to the mitochondria, where it will cause a bioenergetic phase. The mitochondria will stop generating ATP. It will generate huge amounts of reactive calcium species. This will basically lead to the death of the mitochondria. Will then release some factors into the cytosol, such as cytochrome C and H, which contribute to the death. So when I came here to the NIH, I wanted to study the process of how this mitochondrial dysfunction affects other interesting processes such as inflammation. And interestingly, believe it or not, what happens with inflammation is very similar to what happens with neuronal dysfunction. And in this case, we have a monocyte or macrophage instead of a neuron. And instead of having glutamate that activates the cell, we have different inducers, such as, for example, a pathogen or particles, such as cholesterol crystals. And whenever we have these signals, it generates ionic imbalance, similar to what happens in neurons. There is a huge influx of calcium into the cell, which travels into the mitochondria. There is also potassium defects, and all these things cause basically the same thing as the glutamate toxicity. There is mitochondrial damage, loss of memory potential, loss of the capacity for mitochondria to synthesize ATP. There is again a huge increase in back influx into species, famous oxygen, and mitochondria loses the capacity to synthesize ATP and releases also mitochondrial proteins. In this case, mitochondria releases mitochondrial DNA to the cytosol of the macrophage, monocytes, and this mitochondrial DNA binds to a protein called NAD, which is short. NAD, when it binds to mitochondrial DNA, it polymerizes and forms a big complex called the mitochondria. And the function of this complex is to activate caspase 1. Caspase 1 performs the two functions. One of them is kill the cell. It starts killing different proteins in the macrophage or the monocytes, leading to the cell. And the second function is to activate two cytokines, IL-1 beta and IL-18. These two cytokines are very important pro-inflammatory cytokines, and they are released into the extracellular space when the cell dies. What they do is to recruit other cells from the blood, like neutrophils, eosinophils, basophils, into the site of the lesion. So very interestingly, this inflammasome is involved in a lot of neurodegenerative diseases, such as Alzheimer's disease, Parkinson's disease, amyotrophic lateral sclerosis, and stroke. Interestingly, blocking this inflammasome has been shown to block, or at least mitigate partially the progression of these diseases. As we saw in the first talk, with this disease, we not only have like a primary site of the lesion, but also a secondary site, which is sometimes generated by inflammation. So by blocking inflammation, we can block the progression of the lesion, which is very interesting. Another interesting thing about this inflammasome is that, as you can see, these diseases are not really associated with an infection. So this is the process that we call sterile inflammation, meaning inflammation without the need of an infection. In this case, an infectious material that can cause inflammation. And we call the molecules that cause inflammation, we call them DAMs, which means damage associated molecular factors. And other diseases where these inflammation is indicated are, for example, diabetes and insulin resistance, obesity and metabolic syndrome, atherosclerosis, asthma, and ischemic perfusion. All of these diseases are associated with nutrient excess. Diabetes, obesity, therefore, all these associated with excess of nutrients. So we wonder whether we could reverse this process by restricting nutrients. If they are associated with a lot of uptake of nutrients, similar to Western diet, maybe by restricting nutrients, we could basically reverse it. It can be published, actually, in terms of fasting, for example, eliminates the function of eating is going to stop, so we can be sure. And I'm sure you have all heard everywhere, miracles of fasting means prolong your life, living a healthier life. Fasting is everywhere that we see fasting, fasting is always linked to a family of proteins called sirtuins. Sirtuins are proteins activated by calorie restriction and depleted by high fat Western diets, right? And they have been associated with longevity, especially one is the most studied one in humans. Sirtuins basically activate several pathways, that's, for example, antioxidant pathways, oxidation, as opposed to stimulate. So we wonder whether sirtuins would be involved in this cumulative function of calorie restriction. So basically, the aims of my project were first one, can nutrient restriction inhibit function? Can we reverse inflammation by restricting nutrients? The second would be, if this is true, what is the mechanism of distribution and sirtuins involved? And the third one, can we really fasting in a pharmacological way? Because no one has to fast, right? It is one of the things like so. If we can find a way to make this with the drug, everybody will be very happy. So to test this, we developed a clinical protocol at the NIH, and this clinical protocol is as follows. So the patient comes the day before it's pregnant, and we give them a small meal morning, and then the patient fasts for 24 hours. They are only allowed to drink water, nothing else. The following morning, they come again to the clinic, and we take blood from them, and isolate leukocytes. Then we give them another meal, and 1 hour and 3 hours after the meal, we take blood and isolate leukocytes. And then we study inflammation with leukocytes. Well, first we want to make sure that the patients really fasted, because we are giving them $100 for the protocol. That's a waste of money, right? So we test this basically by checking the insulin levels. Insulin levels must be low after the fasting, and then go up to the eat. Glucose levels are low in the fasted state, then goes up to the feed, and then growth hormone levels are high during the fasting and go down after feed. We were lucky because all the patients complied with the protocol. And then basically we showed some results. We found that it's true that fasting increases inflammation of human leukocytes. We checked 3 inflammatory cytokines, IL-1 beta, IL-18, and TNF. And as you can see here, we checked 19 patients. Virtually none of them in the fastest state, the release of cytokines is lower than so this is fasting. Then we wanted to know the mechanism of this inhibition of inflammation by fasting. This is the only complicated, that's why we don't call it drug. But basically, we see that fasting increases mitochondrial function. So you can see here, respiration in the fasted states is higher in the than in the fed states. Both basically, we stimulate the leukocyte activations. Also, reactive oxygen species are lower in the fasted state compared to the fed state. I will skip this for a second. Then we check the activity of mitochondrial protein cholesterol tissue peroxide dismutase. The function of this enzyme is to basically destroy these reactive oxygen species. And consistently in this data, we find that the activity of this protein is higher in the fastest state than in the United States. This is because this protein is activated by SERT3 on the SERT2. So we wanted to see whether SERT3 was involved in this pathway. So we eliminated expression in cells or overexpressed. We can see that when we knock down SERT3, we get an increase in inflammation. When we overexpress it, we get an increase in inflammation. So this is SERT3 is involved in this pathway. Basically, these are other results that we have as a model, which is that fasting increases the activity of SERT3. SERT3 increases the activity of SOD, which is the enzyme related in increasing these levels. All these together with this improved respiration leads to a better mitochondrial quality, and as I showed you before, mitochondrial function really determines whether the cell dies or lives, whether there is inflammation or not. Finally, the point where we wanted to find a drug to manipulate this right instead of having to fast. We found this compound called nicotinamide riboside. It is a precursor of NAD. NAD is related with vitamin D3, and it is known to be an activator of SERT3. So we wanted to, instead of fasting the patients, give nicotinamide riboside to the cells of the patient and see whether we could mimic the effects of fasting. And it's true, we can. We can enlarge the cells. There is biomorphs in the fasting state, and also the administration of NR increases the rate of biomorph in the cells. And so, basically, the results we are trying to, we are developing some clinical protocols where we are using both fasting and this compound NR to try to increase inflammation in actual patients. We are using our protocol for MASH. I'm trying to use fasting to decrease inflammation in those patients, and we are also in the process of developing a protocol for the use of NR in patients as well. So the summary of the story of inflammation is activated by mitochondrial dysfunction. The sterile inflammation is associated with diseases related to nutrient uptake, such as diabetes and atherosclerosis. Fasting decreases inflammation by increasing mitochondrial function. Nicotinamide riboside treatment limits the effects of fasting on mitochondrial function and inhibition of inflammation. And inflammation is also associated with consistency. This is a way of regulation to reduce as a way to activate those diseases. And we'll find a way to take the collaborators, and so this that hasn't been involved with inflammation until lately. But after my studies, a subgroup of the lab has started studying neuroinflammation. So you, Cardian, and Kohuli are studying the effects of inflammation in Parkinson's disease. We have a mouse model of Parkinson's, Parkinson, and they are trying to describe what's the inflammatory consequences to Parkinson's disease. That's it. Thank you very much.
So you have some questions. We have some questions. Yeah.
Congratulations. Can I speak Spanish or Spanish? Okay. Congratulations for all of us, for all of you for the talk. I would like to. I have some questions. I would like to start with Javi because it's the happiest. So it's very interesting that what you explained. But you said that the patient eat food increase inflammation. Yes. But you said that the fasting increase in the activity of CO2. Yes. Increase the activity. That is. What kind of food? Because it's just eat, or you usually you prove with different kind of.
Basically, we gave the patients several options. We had a vegetarian option, we had another option that had eggs, pancakes. We saw the same effect with all the different food
Even
options.
with the with vegetables.
Yes.
With the patient in vegetables, use the inflammation.
Yes.
Nice.
And they look at the basis, if they're all normal weight.
Yes, we chose BMI between 22.5 and 26. So it's normal. Patients regular weight, normal weight.
Trying to eat to get
Yes.
Because 500 calories by itself is a huge meal. No, when you think about something like that, you tend to think about inflammation caused by huge amounts of overeating and things like that, and rich foods. And so, to me, that is more of a just a regular response of your body increase. It's an unusual thing that you see it regardless of what happens. So, what do you think is the difference between what you're seeing and more of a pathological effect?
Well, I assume that in the pathological way, that all of these markers would be really much exacerbated. This is like a very mild inflammation. The increase was not really very big. It was consistent in all 19 patients, but it's not really huge. Another thing that I haven't explained because of the time limit. In fact, we measure LPS. LPS is a molecule present in the wall of the bacteria, right? So we measure LPS in the blood before and after eating. Eating by itself increases LPS in the blood, suggesting that the food somehow increases the permeability of the gut. And the bacteria that you have in the gut are taken, or at least the LPS, which contributes to the inflammation of the disease, which is fascinating.
That you think that they cause the bacteria. And we get inflammation because we don't wash well the vegetables.
No, you have to
No, it's just that the bacteria that they have, there is, if you remove all bacteria or this from the food, you will want to reduce the inflammation.
I don't think that matters. It's the bacteria that you have in the gut. Just,
Okay. So it's just for you know.
Do you have any idea what BMI process changes your needs? Is when the BMI will be able to see any
yeah.
party actually qualified by some certain. And so I was wondering if you guys have to get
We haven't checked that. Interesting. Once we have the clinical protocol to study these patients, we can do so many things.
So you have to look up leukocytes, how to look up other cells. So it's the hard to put in a model like
in the patient, we have only used PDMCs, whole and monocytes. In mouse, I've used bone marrow macrophages, I've used brain macrophages. So with peritoneal macrophages, you can study the effect of fasting. Because you fast a mouse, you take the cells from the peritoneum. With bone marrow macrophages, you cannot study the effects of fasting on the mouse because it takes 1 week to differentiate it. So the effect of fasting is lost, right? But it would be interesting too. So.
I will continue with my question with Sonia. You said in your talk that you know that you treated this marshal brain damage with melatonin, and the melatonin have protective effect on the brain. Do you think that it's possible that this effect is because the antioxidant properties of the melatonin or any other reasons?
Yeah, so it's partially related and immune proliferation in some way. It is glial that are white matter, so we don't like more lymphocytes, and that's me more mind in the white matter. That's mostly in in vitro study that you have in the form this in the mind. Yeah, but there are other problems for things. But again, the fact that was not the same in the other animals. So you have in the brain. It's completely dependent.
other protective effects for renal, brain for the inflammation, brain inflammation. This effect can be for the estrogen.
Yeah, I don't know exactly. I think that there are it's individuals for why there are this kind of difference, but also there are studies that show that the protective mechanisms that there are with inflammation they come through different pathways and the reason for the bond some difference in the neonatal animals, in the neonatal animals will have estrogen. So, yeah, I don't think that is yeah, but then there are I have that. I have a question for you, your work is very exciting, it's very interesting. So the way that you can visualize the tumors in the technique that you're doing, you think that can be in humans in the future can be applied this technique like some kind of methodology that will be easy for the nurses determining which is the healthy brain, which is the
Yes, we think so, and we hope that so. First of all, because we put together, not me, I'm not part of the patent, but the PIs put together a patent for this work. Second, because OCT is already FDA approved, but the ophthalmologists have been using it before that for a lot of years. Third, because there's a microscope that already exists for the ophthalmologists when they do retinal surgery. So the only thing that we should do is have more data from patients. We did already 3 patients. We are putting together an IRB protocol to do it in at least 30 patients at Hopkins. And next step is trying to get the companies maybe looking, but hopefully we are going to have it sooner than later. The second part that I'm not sure was of interest at that time is we are working with biomedical engineering in all these ways to look at the brain during the surgeries. And the second technology is with photon that I'm sure that a lot of you use for different experiments. And the OCT has a larger field of view, so it kind of gives us those maps with a red and green that we put together. But then the photon has a much smaller field of view, which means that it's giving us a histological picture. And what we are trying to do, and we are putting together another one to try to get funding, is to use that during the surgery to have an intraoperative diagnosis. So instead of getting a biopsy, sending it to pathology, waiting for 2 weeks with the stain, time and chemistry, just having the picture right there, bringing the pathologist into the operating room and having the diagnosis intraoperatively with the hopes that in the future we are going to have personalized therapies that we could give right there. So we are trying to put together these different pathways to improve that treatment. That's longer in terms of timeline because the program is not actually approved yet for patient use. But we are trying.
There are, I don't know, this confusion methodologies similar in clinical practice, right?
So for the OCT, there are several groups that are trying out with BREF, but we have one of the engineers that comes from the MIT with the optical expert in the one of the optical experts in the world. For the multiphoton, actually, just 1 month and a half ago, team from Germany, because in Europe everything is easier, you don't have to go through the FDA. They tried multiphoton in brain for the first time. So there's a little bit of competition in between the groups. Yeah.
Good. Thinking of the lines of deciding laser guided high dose radiation delivery system that will actually kill the basis of the imagine that we get
Yes.
out the cell with high dose radiation.
That's
Something like standardized style or something?
Yes, that's a very, very good question. And that's what we are thinking too. So the same engineer is working also with GI. So they already have, with not with OCT, because OCT, the good thing is that doesn't damage the tissue. But multiforon is so strong that one of the drawbacks is that for imaging is that it produces what we call photo bleaching. Like in the pictures, if you expose them too much, they get burned. Same thing with the tissue. So we're thinking if we could use that as a benefit and with a low dose get the imaging and with a high dose destroy that tissue. And they try to do that already with GI. The problem with the brain is that when you have something like that, the brain is so sensitive that you can produce a seizure. With heating, with strong light. So we may get a little bit of resistance from FDA and all these regulatory entities. And we have to say, we are trying, we will try, but it's a very, very good question, of course.
And we have to understand about the person who is in it, that I don't know whether markers or themselves does not use follow up.
Right. So the good thing with optical coherence tomography compared to other technologies that already exist is that we don't use dyes. We don't have to give anything to the patient that makes the tumor highlight. Okay, so the only thing that the OCT uses is how the different tissue reacts to the wavelengths. So, and we think that what makes the difference is a myelin. The amount of fat in the myelin in the normal tissue versus the cancerous tissue. So that's a very good question. What we found is that the cancer versus the normal tissue has different optical properties. We call it optical attenuation maps, not the color ones, the ones that I showed that were black and white. So what happens is that in cancer, the light, against what we would thought, can cross better, because in non-cancer, the myelin, it's where the lipids change how the light reacts. So we had to find first that difference exists. And after that, what we did is we calculate the sensitivity and specificity, and we found the first code that separate the number of optical attenuation. All those are numbers. So normal tissue has an attenuation of 7, cancer has an attenuation of 3. And then what we did is we created algorithms. So instead of having the surgeon looking at those numbers and saying, oh, what the hell, what am I going to do here? That the machine, the computer, translates those numbers in a map, in a 3D map that is green or red. But behind those maps, that is like just making easier for the surgeon, there's all these numbers. So every single green dot is a 5 or a 7 or a 3 in optical attenuation. Did that respond to the question?
But I really love your and I just also for the connection.
Yeah, it is some of the applications that we use exactly understand how the brain really on in my lab is like a big effort in neuroscience, but now that in the human context and trying to understand how the human brain responds to actually detect this is further activity in different types of diseases. And you can actually damage the brain and try to understand the toxicity there and understand how the brain is going to adapt or the same is that like animal models of using a disease for like physical damage.
That's where we have the back.
Yes.
Yes.
That is more related to the treatment.
Okay.
I work in the field of mesenchymal stem cells. So you were mentioning that one of the treatments that they are using now is the use of mesenchymal cells that have been threshold with one more organic factor 4. It deters progression of cancer cells. Right. I am pretty much aware of the stem cell and differentiation of mesenchymal, and when you talk to a surgeon who does the same thing that we have, and what he's telling me is that unless you use your mesenchymal cells delivered within a matrix, by a matrix I mean connected tissue with collagen, with growth factors. In the past 5-7 years, it has been found that just administration of mesenchymal stem cells without that support does work.
Right. So that's also a very good thought, because one of the first experiments that we did, first of all, is to see if we could keep them as mesenchymal stem cells. And the good thing of having a neurosurgeon is that for our pituitary surgeries through the nose, once you take the tumor out, you have to make a small incision in the belly to use fat from the same patient as a plaque. So the brain doesn't come out through the nose right after the surgery. So we use the same fat from our patients to extract those stem cells. And the first thing that we wanted to know, why are we using stem cells? For two reasons. Number one, because as you know, they track inflammation or they track damage. So we found that they track the cancer. So we want to use them as a Trojan horse that carry our therapy to where the cancer is. But what happens? The first thing we did is in mice, we tried to study how many stem cells would get to the brain if we injected them in compared to different pathways. So we did intracardiac injections, we did tail vein injections, so venous injections, we did local therapy, and then we did suprarachnoid injections. And what we found is that the most efficacious way is doing local. So another of our lines is working with biomedical engineering to see how we can do those local therapies. And we are using nanofiber hydrogels to try to have the cells in there. And that's where it's so important to have the intraoperative diagnosis. Because what we, in our dream life, what we would like to do is have the patient take the tumor out, look at the tumor, have the diagnosis there, harvest some of the mesenchymal stem cells the same way that we are doing already with our pituitary patients, mixed with whatever pharmaceutical products that we find that can fight brain cancer, mix that with the fat cells of our patients and inject that with the nanofiber hydrogels in local therapy. And local therapy has been explored for a while in brain tumors already, because why? The brain barrier is very difficult to cross. The stem cells can cross it, but I think it's only 5% that can get in. And the chairman at Hopkins was the first one who discovered one of those 4 FDA-approved drugs, and he did wafers of carnosine with biomaterials, and after taking the tumor out, they put the wafers there and they deliver local chemotherapy. But there's the number one problem is that we don't have a drug that kills that cancer yet. But we are trying to advance in other ways to see how we can do it.
So most of the people from part of the eye, the brain, which is calm. So you said it's non-invasive, but you showed us.
Right, because you use it anytime that you have to do a surgery. So non-invasive means that the laser by itself doesn't burn the tissue. It doesn't even have to touch the surface, because the light, compared to the ultrasound, we are already using ultrasound to see where the tumor, because sometimes when the tumor is not in the surface, but we use the ultrasound, but the ultrasound
has to touch the brain. This one is not, we call it non-invasive because we don't have to administer that. It doesn't touch it, but it cannot cross the skull. But usually we want those patients into a surgery to do, yes. Yeah.
And you said, I understand correctly, the brain spires as a vector can go from one cell through synapses to other cell.
Yes, yeah, that is how the last day is biology.
But then it's moving further.
No, it doesn't because of the modifications that they are introduced in the brain virus. And this single type modified virus, so it's like it's been attenuated. The regular case, it will keep jumping, and they will actually defend like very clearly distinct neuronal sequence. You know, it will keep going and it will never stop. And it will create problems.
Do you have any differences?
Yes. Yes, yes.
The ECUSA is exposed to the virus is now neuronal. So in the virus. And so when we go to the experiments for neuronal
Well, you mean for the yeah, I will actually not be involved in that. So it's some group in the lab that is going to do neural inflammation. There is the Parkinson's model. Yeah, there is in the Park2 neural mouse. But they're mostly right now doing stuff in vitro neurons. You know, they haven't done much to them. But actually, we had a protocol with patients of Parkinson patients. And somebody in the lab studied also inflammation. They saw actually a decrease in inflammation in the SPARK2 mutations, which was very fascinating. Still an increase in inflammation. So I think that project was kind of fun.
You actually have a joke. You plan to check if you don't eat, it's probably gets stressed. If you don't eat, yeah, you're gonna get stressed. Probably. I know. You're gonna get that. I'm looking at those. I'm getting stressed already. How can you measure? That's basically what I'm measuring. That kind of influence of that stress on the immune system of the individual, and therefore the production or not production levels of lymphocyte T or lymphocytes or different kinds of. I know it's a joke, but I don't think it's a joke.
No, we should include as control.
I mean, there are all kinds of things that can change your populations of lymphocytes, different types, different CD families. Not eating for 24 hours are induced to eating on a regular basis. Probably changes something.
Well,
Yeah, we didn't try that because it would be more complicated, you know, for the protocol perspective, right? But yeah, 24 hours. The patient, when they came to the clinic, they were devastated. Some of them almost fainted when we took the first blood. Between 22 and 38. No, they go home. So we have to